Webinar is now live.
How are people raising hands?
Okay, we're live on Facebook.
Okay, I'm gonna stop sharing the screen. Nyaradzo, can you come on camera, please? Oh no, you'll come when we call you. Sorry, it's fine. I'll be the one who's on camera. Okay, 1 more minute. I'm starting the countdown. And we can let everyone in a second. Good afternoon, good morning, good evening, whatever time it is in your side of the world. My name is Sponsor Pilane and welcome to the Global Lecture and Engagement Series for supporting community and civil society engagement in the IAS COVID-19 Prevention Conference. The African Alliance, the Vaccine Advocacy Resource Group, and the Global Network for Young People Living with HIV, as well as Ready Movement, bring you this series of lectures focusing on the 4 pillars that will be discussed at the International AIDS Conference's COVID conference, which is under the theme Prevention. Before I go any further, my name is Sponsor Pilane, and I have the pleasure of facilitating these lectures for you. As I said, the IAS conference will be hosting the COVID-19 Prevention Conference, and this will feature the latest in prevention-related science, policy, and practice. This conference is going to take place virtually next week, Tuesday, and will also include invited speaker sessions as well as abstract presentations. And in order for communities, particularly civil society, who have been at the front line of the fight against COVID-19 and the pandemic at large, the objective of these skills building sessions is to maximize an engagement and also ensure that community voices are able to take part in the IAS conference and are able to meaningfully contribute in the ways that are required. And to bring us the first lecture today, but before we even get there, I'd first like to welcome the head of the African Alliance, Mr. I would like to welcome Tian Johnson, the head of the African Alliance, for them to give us a bit of a welcome. Tian, over to you.
Hi, Ponso. Thank you so much for that introduction. Hello, everybody. A very warm welcome from us at the African Alliance, the VARG, Y+ Global, and the Ready Movement. As Ponso said, we're super excited to have you here, and we're really keen on breaking down barriers and building bridges between communities and accessing conferences like the IAS conference that, whilst they have really tried to reach out to communities and have various aspects of community activities, have really needed and desired to be more in touch with communities. And so in partnership with our colleagues, we bring you this series of 4 lectures, and we hope it will be 2 days of learning, of engagement, and of deep reflection. And we really just wish you well, and we look forward to an exciting 2 days of engagement. We encourage you to engage, ask questions, ask comments, hold the lecturers to account if what they're saying doesn't resonate with you, or you might want to unpack it a bit more. And again, welcome on behalf of all of us, and we wish you well over the next 2 days. Thank you, Ponso.
Thank you so much, Tian. As I said right now that we are going to start, we're about to kick off the first of the 4 lectures. And before we do that, I have shared a link in the chat where we are asking you to please fill in a pre-lecture survey. And this survey, it will help us to determine the amount of knowledge, your expectations, and it also help us to make sure that we are meeting your expectations. We also have a very special treat to those who do fill in the survey. If you fill in today's survey and the survey that will be shared with you tomorrow at the end of the lectures, you stand a chance to win 1 of 10 free passes into the IAS conference taking place next week. You would have to fill in both surveys to stand a chance. The winners, the 10 winners, will be selected randomly using an electronic randomizer and selector, so there won't be any kind of human intervention from our side, and we will ensure that you're able to fill this in, and you will be let known, or we will alert you via email if you are a winner. I have reshared the link again. There you'll see it. It's a Google form. It fills in like most surveys, and then you just submit, and you should be able to fill it in. In the meantime, while we give you a few seconds to fill in the survey, I know we are multitaskers in the world of working from home, so we are able to do more than one thing at the same time. Bringing us the first lecture of the series is Dr. Nyaradzo Chigorimbo-Tsikiwa, and she is a senior researcher in the Department of Medical Virology at the University of Cape Town in South Africa. She heads up a small research group which investigates the interaction of viruses with the human body, especially looking at ways to prevent infection. Before this, when she was still in the early stages of her career, she was a research fellow also at UCT and the Desmond Tutu HIV Foundation. She completed her postdoctoral fellowship in 2013 in drug discovery, and in which she was part of the multidisciplinary team that developed the first African clinical candidate, which was developed using modern drug discovery paradigms. She is passionate about homegrown African solutions and interventions to tackle African problems. Nyaradzo is here to also currently uses multidisciplinary biomedical approaches to understanding and finding solutions to prevent HIV. She works with several international collaborators and has presented her work internationally to scientific conferences and forums, as well as to the public. With a warm welcome, I'd like to welcome our first speaker, our first lecture. This is Nyaradzo. Dr. Nyaradzo, please welcome.
Thank you very much, Ponso, for that warm welcome. I hope you can all hear me. Okay, Ponso is nodding her head, so I'm sure you can all hear me. I'm very excited to be part of this. This is one of, I'm used to presenting to scientists most of the time. So this is the first time that I'm presenting to people who are not core biomedical scientists. So I'm very excited about that. And the style of presentation that I prefer is very interactive. So if at any point I say something that you don't understand and you'd like me to elaborate on, please feel free to ask me that. I think we have a chat that's ongoing, and so you could put your questions in the chat box there, or raise your hand up and Ponso will alert me to any questions if I don't notice them myself. But yeah, I'm really looking forward to this. I'm going to share my screen now so you can see this presentation. Okay, let's see. Okay, oops. I'm wondering if you're able to see my screen.
Yes, we are.
Are you seeing the presentation?
Yes, we are. You just need to put it in slideshow mode.
Oh, okay. Sorry. I think I might have started sharing that too early. Okay, let's do that again. Okay. Oh. For some odd reason, we seem to be having a bit of a glitch here. Okay. Okay. Are you able to see that?
Yes, we are.
Okay, great. Okay, great. Fantastic. Okay, so as Sponsor said, my name is Nyaradzo. I go by the name of Nyari most regularly, and I'm a researcher in HIV. But, you know, with this new coronavirus that has come up, a lot of us previously HIV researchers have, you know, been co-opted into this SARS-CoV-2 work, which has been very exciting. We've seen a lot of science coming up, which has been very exciting. And a lot of, you know, societal changes have happened because of this, I'm sure, which you're all aware. And it was quite interesting to see in the chat box, actually, that someone from Uganda, Emmanuel, I think, mentioned that he has a lot of views that he would love to share and wants to engage with here. So I'm really looking forward to be able to engage with you, community guys, you know, and advocates who are, you know, in the forefront of this pandemic on the ground. And so what I'm going to do today is I'm just going to go through a bit of background on what's happening in the vaccine landscape. I have tried to access a lot of the works that I'll be referring to are really available commonly in the media and are written by journalists. So hopefully they should be accessible to anyone. So what I'll start off by doing is I'll talk about the SARS-CoV-2 vaccine landscape. I'll talk about the process of how a vaccine is developed so that we can understand where we are when we start talking about vaccines. And then I'll briefly talk about the approved vaccines that are there currently. And then I'll get into some basic immunology and biology. And really the reason for this is because when we start talking about the different types of vaccines, there needs to be a bit of background there. But don't worry, I will not use a lot of jargon. I will try and make it as accessible to everyone as possible. And then we'll then get into the types of vaccines that are available. And then we'll talk a little bit about herd immunity, because this is a concept that's flying and floating around everywhere a lot. And then we'll obviously talk about the virus variants, which are, especially in South Africa, quite topical, and in other parts of the world, like the UK. And then I'll just briefly share a few concluding thoughts, around the ethics around human challenge assays and that sort of thing. Okay, so this is an infographic that shows us actually what happens in the process of generating a vaccine. A lot of concerns that a lot of people have raised are, focus around a lot with the speed in which these the SARS-CoV-2 vaccines have come up. I mean, it has been unprecedented, the speed that these vaccines have come up, which is true. I mean, it normally takes 10 to 12 years to actually develop a vaccine candidate, but here in less than a year, we actually have vaccine candidates and some of them already licensed for use in humans. And so it really has been quite fast and, as I said, it's unprecedented, but I think a lot of that has to do with the fact that, a lot of people globally all came together to focus and work on this one common goal, which was finding a vaccine. And so the process is that the steps that normally take place, which actually also took place with this vaccine candidates that we're seeing that are coming up, is in the first instance, what happens is that scientists would come together, like biomedical scientists would come together with molecules that they think would be able to confer activity as a vaccine. So a vaccine is anything that can provide immunity towards a specific disease. And so there are various things that can do that. They're either proteins or RNA or even DNA vaccines that can provide immunity. And for all of those different types of molecules that can be used in vaccine design, they all obviously start off, being investigated in the lab, where they are investigated in test tubes. And once you find that, using in vitro assays, that, these molecules can actually confer protection immunologically, then you test them in small animals. Before you can give anything to human beings, unfortunately, I mean, I know it's could be contentious, but before you want to give anything to human beings, you want to test that it's safe. You start off with small animals and move on to more complex animals before you actually give it to human beings to test the safety first. And so that's the first thing. So you start with your preclinical testing, which involves testing small animals and some in vitro assays. And then once you find that your vaccine candidate is safe and works well and efficacious in those assays, then now you move on to the phase 1 studies. So these phase 1 clinical studies are where you now get into human beings. You're now sure that the vaccine is safe. No small animals were injured or had terrible adverse events during the testing stages previously, and then you can now move on to healthy human beings to test whether or not it is safe in those human beings. And normally this is quite a small number of people that you test in, less than 100. And then in this case, you test the dosage because you need to know how much of the vaccine you need to give the efficacy, and also to see that it actually does elicit an immune response in a human being. And so this phase 1 safety studies are really focused a lot on looking at safety and the dosage and immunogenicity. And then once your vaccine candidate has passed that step, and mind you, a lot of vaccine candidates do not pass this stage. In fact, in the very first stage, a lot of molecules actually don't pass through this stage. Just to give you a rough idea, with the SARS-CoV-2, I think we had over 350 clinical candidates being tested, but we have about 66 that have actually progressed onto stage 2. And so stage 3, which is known as the phase 2 or expanded clinical trials, this is where you now get into hundreds of people. And in this study, you split the participants into different groups. This is to measure their safety. So you would look at it, perhaps you want to look at people who are elderly, and in this case of SARS-CoV-2, that is the case because it affects disproportionately people above a certain age. So then you would have a group of people who are older, and then you have a group of people who are younger, who you want to test whether or not it's safe in both the elderly and in the younger population, and still really focused on safety as well. And then once you found out that this vaccine is safe in the elderly as well as the young people, which is what we've seen, then you now move on to the phase 3 studies, which are the efficacy studies. Now, this is where you now bring the study into thousands. In this cases, for a lot of these vaccines, they've been in 70,000, 40,000 individuals already. And this is where you now look at the efficiency of this vaccine. And in this case, I see Wilson Zulu has raised his hand. So I'll let you take that if there's a question. I don't know, do you want him to unmute or should I carry on?
I'll get to it. I'll tell you just now. Allowed to talk. Wilson, you can go ahead.
Great, thank you. So in the phase 3 efficacy studies, what we normally do in this stage is you then separate your participants into 2 groups where you have, and in most cases, the process is blinded, which means that the researcher themselves and even the participants themselves don't know whether or not they'll receive the vaccine or a placebo. Efficacy studies do not give the virus itself to individuals. So there's a lot of talk going on in the media that if you participate in clinical trials, you'll be given the vaccine. That is not correct. You do not give people the vaccine or the disease agent during efficacy trials. What you do is you give a group an arm of the individuals in this study the vaccine itself, and then another arm you just give them a placebo. A placebo in this sometimes is saline, which is just normal salt, or sometimes you just give them you know, the adjuvant that the vaccine is also part of. But in no cases do people actually get given the virus itself. We will talk more about this towards the end when we talk about human challenge studies, but phase 3 efficacy clinical trials do not involve giving human beings the virus itself. And so in this case, this is where you then split up your participants into 2 groups, as I said, and then you assess in your 2 arms which has the number of people that naturally contract the infection. So what they would do, so as for example here in South Africa, where 3 clinical candidates have actually been tested, for example the Oxford vaccine candidate, they would have a group where they would give them the vaccine and then they would let them go out and carry on life as normal. And because the epidemic is raging on in the country, you would expect that a certain percentage of those people would get infected, and then you take the difference between those who are infected, who have received the vaccine, and those who are not, and then you can, that's how you now check your efficacy between those 2. We'll speak a little bit more about that later. And then this is also very importantly the stage where you actually assess your adverse events. Your adverse events are very important. And in this case, you want to make sure that you have numbers that are large enough to be able to assess if you have rare adverse events that take place. So for example, if people get rashes when they get this vaccine, if people get Bell's cerebral palsy, as people allege, this is where you would see things like that. You would give the vaccine to 20,000 people, and you know that in 20,000 people, if somebody is going to get an adverse event, that's a number that's high enough to see that. And so all clinical studies in phase 3 are powered enough to be able to see these differences. And then once everything goes through the efficacy stage, you find that your vaccine is efficacious and there are no adverse events that are lethal, then you can carry on to the next stage, which is the approval stage, when your vaccine is then approved. Sometimes, as we've seen with the SARS-CoV-2 vaccines, we get early or emergency approval, where because of the pandemic, we have said, when we've seen vaccines that are safe, we say we can start to use them already because we've seen that they're safe and they're efficacious. I think I could take that question in this stage before I move into the next stage, Ponso.
Wilson, go right ahead. You can unmute your mic.
Good afternoon, Dr. Nyaradzo.
Hi.
Yeah, I wanted to find out when you try to roll out the vaccine on human subjects. There is a question that has been running on my mind for quite some time. I'll talk in relation to BCG. BCG, it's, when one gets a BCG, it is latently infected by TB, but you cannot pass it on. Now, in this regard, we're looking at the totally new disease, that is the COVID-19, and if at all the vaccine will be introduced into human subjects, would we comfortably say such subjects would be also latently infected by COVID-19? And if so, those people with the whose immunity is compromised, giving an example of people who are HIV positive, what are the parameters that should be taken into consideration before actually administering such a vaccine on people like those, and those with underlying factors, say for instance, those with the polycystic kidney disease and the chronic kidney disease? Yeah.
Yeah, thank you very much. That's a really good question. If there's another question, maybe I can take that and then I could answer them both at the same time, if possible, or else I can just respond to this one at the moment. Ponzo, you can guide me.
There is another question. Just hold on. Margarita, you can ask your question.
Hello, good afternoon. I would like to know if someone who has got COVID-19 before will need to get this vaccine or not. Sorry about my English, I'm from Mozambique. I would like to know that if it's in some way, it will be an obligation to get the vaccine for example, someone who is at the risk, in the group of the risk group, if it will be an obligation to get the vaccine in some way.
Okay.
Thank you.
Okay, cool. I got those questions, and those are really, really good questions that you've asked, and they are obviously very important to a number of individuals. So the first question, I will speak a bit more about it when I actually speak about the types of vaccines that are there. I will get a bit into the mechanisms, actually, which will, I think, give you a bit more information there with regards to, do we actually latently infect people with the coronavirus? The short answer is no, we don't. But sometimes maybe a little bit, so it's not very clear right now, but that will become clearer now exactly when I explain a bit later. And then the question on whether or not people who have had COVID should get vaccinated again, that's a really very important question. And the current agreement right now is yes, and I will also allude to that later as well. And the reason for that is we found that people who have had COVID infections before do not necessarily have immunity that can prevent another infection. It seems to be possible that people who have had COVID can actually get reinfected with the different variants, but we'll speak more about that later. And I see there's some more questions there, but I will just move on for now, and you guys can type your questions in the chat room, and then I will also get back to those. Okay, so the current situation right now with the vaccines that are available, as I was saying earlier on, we've had over 300 vaccine candidates that have been tested. We've had 66 which have gone into clinical trials, which is a huge number, to be honest, actually, but which is very good. And the reason for that is that a lot of people, I mean, thousands and hundreds of thousands of people have actually come together to work on these vaccines. It's been an incredible effort, and the work has actually yielded a lot of results. And so what we have is, if you remember those phases I was speaking about, currently, and this was data that I got on the 26th of January, and this information is available readily online, and you'll see some of the references later. We have 38 vaccine candidates in phase 1 right now, which are still being assessed for safety. We have 24 that are actually in phase 2, and in phase 3, which is the stage where we're looking at dosage and adverse events, we actually have 20 clinical candidates, which is really good. But we have 8 vaccine candidates which are already in limited use, which is a large number actually. Unfortunately, for a lot of us in Africa, we haven't seen those yet, but that's a discussion that I think we'll get into a little bit later. But we have 2 vaccine candidates that have already been approved, and we have 3 that have been abandoned. And those ones that are abandoned because they've been shown to be not efficacious, or they have adverse events that are not acceptable. And so of all these, there are still 90 that are actually currently undergoing tests in preclinical in preclinical assays, so in animals and that sort of stage. So we have a lot of molecules that are being assessed for that. And this sort of gives us a summary of that, as I was talking about. And all of those numbers represent different types of vaccine candidates. So we have the RNA vaccines, we have the DNA vaccines, and of these RNA vaccines, we have 29 that are still in the preclinical stage, but we have 2 which are currently in use. And these ones are the Pfizer and the Moderna one, which are currently in use. We'll speak a little bit more about them. And then we have the DNA vaccines, as well as the vector and replicating and non-replicating ones. And then we have the inactivated viruses, which I think the earlier question refers to. And then we have the protein subunit vaccines and the virus-like particles. And we have a couple which are unknown because these are still in preclinical stages. And so, I mean, you can see we actually currently have quite a lot of candidates that are in use, which is not a bad thing. And so this is sort of what the landscape looks like. I know this slide is quite busy, but the reason why I brought this is just to sort of let you guys actually see and know that there is actually a lot of work that is going on behind the scenes. When you see an announcement about a vaccine candidate that is actually being approved, there's a lot of work that is happening behind the scenes. As I was saying, there are many, many candidates that are being tested, and there are different institutions that are doing so. So for example, in these blue ones here, this just shows the genetic vaccines. There are over 60 candidates which have been tested. And so by the time we get one, it's one of many that has been approved. So there is a lot of work that is going on. Of these that are being tested, we have a couple that are the leading ones. And the leading ones, in this case, is the one that everyone knows most about, is the Pfizer one, which is an mRNA vaccine. Now, this one has been approved in Saudi Arabia, the Bahrain, Switzerland, and it's also being used in the US and in the EU, as well as other countries. And then we also have the Moderna vaccine, which is also an mRNA vaccine, which is currently in phase 3 studies, but also has emergency use approved in the US, the UK, the EU, and other countries. As you will see, a lot of these vaccines are approved in the North predominantly, and are being used in those instances. The Gamaleya vaccine, this is the Russian one, which has already been licensed for use in Russia, and other countries have followed suit with that. And then the Oxford AstraZeneca vaccine is one that has also gone through clinical trials here in South Africa, as well as the Pfizer BioNTech. So a lot of these have actually gone through clinical trials in Africa, which a lot of people have had different views on, but from a scientific and biomedical point of view is actually a really good idea. The reason for this is that we know that different populations of people have different responses to drugs, and the vaccines could follow suit. So, for example, we know from the ARV field, for those of us who work in HIV, we know that some of the dosages that certain ARVs were being used were actually too high for people of African descent who have a different makeup, which results in them not having certain enzymes which actually digest these drugs. And so as a result, a lot of individuals in Africa were having the drug in very high concentrations, which were quite toxic for them. And the reason for this was because when those drugs were being tested, they were not tested in African populations. And so one of the good things that has happened with these vaccines is that actually we've had African people participating in them. So we have clinical data on how Africans respond to this. So these studies have taken place in Africa, in Europe, in the US. And so we know that how people respond from different ethnicities to these vaccines, which scientifically is a very good thing. And so we we also have other vaccine candidates. So, a couple of them which are being developed in China, which are also being used, like the CanSino, which is being used in China currently, and then also the Johnson & Johnson vaccine, which I think perhaps in the global south might be a vaccine that might be an option for us because of its storage requirements. I think a lot of us are aware that, for example, the mRNA vaccines like the Pfizer BioNTech requires storage at -80 degrees, which is practically not logistically feasible for a lot of our places in Africa, and just increases the costs really tremendously. But we have other vaccine candidates that do not require such stringent storage facilities, and that is mainly because of the type of vaccine that they are. And so depending on how the vaccine was developed and what type of vaccine it is, the storage is also different. And so the first ones that have come out, these mRNA vaccines, unfortunately, are the ones that require those very impractical, in some ways, storage conditions, and as a result, might be challenging for us in the global south. But there are other options that I think are available, which will come up, which will probably be more practical for us. And so perhaps I could take some questions at this stage before I move on to how these vaccines work themselves. Ponso, I will be guided by you.
Yes. Gali Chauluka has a question. Says, how long does it take to monitor these small animals and initial human subjects before it is approved for human use? Another question, Doc, that came very much earlier, also from Wilson. I'd like to know what effect the vaccine would have on people whose immunity is suppressed due to HIV and those with underlying conditions like CKD. What other parameters? What parameters will they need to look at before giving such people the vaccine? Would you like another question?
Let me take those two for now.
Okay.
So, yeah. So, you know, the second question really also speaks to, you know, some of the necessity for vaccine trials being conducted in different parts of the world. So here in South Africa, for example, or in Africa, we have a huge population of individuals who are living with HIV and are taking ARVs, and we have a high prevalence of TB, for example. So when clinical trials were done here in South Africa or in other parts of Africa, a lot of participants who were recruited were actually individuals who are living with HIV. And so I know for a fact that the Novavax vaccine, as well as the Pfizer and the Oxford AstraZeneca vaccine, had a cohort in its phase 3 studies of individuals who are living with HIV and was conducted in a TB endemic area. And so we now know, you know, from that data, what people who are actually living with HIV, those who are on ARVs, and in some instances, those who are not, because they were also recruited in the studies. So we do know for a fact that people who are taking ARVs, those vaccines are safe because they've been tested in people who are living with HIV, who are on ARV treatment. And we also know that these vaccines work in people who are living in areas with high TB prevalence. A lot of these, for example, the Johnson & Johnson one also, as well as that Oxford AstraZeneca one, as well as the Novavax, was tested in Cape Town, which is a highly TB endemic area. So from that data, we know that they are safe in individuals like that. So I hope that answers your question. With regards to other specific diseases, it will depend on the specific vaccine trial to see if they had enough participants who had those diseases. But I think in a lot of them, unless specified, they might not have had enough numbers to have that. But we definitely know for people who are living in HIV what the safety of the vaccine candidates is, as well as people in high TB endemic areas. And the question on time. So before the phase 1 studies typically take about a year, you know, to finalize, to see what the safety is. In the case of the SARS-CoV-2 vaccines, that actually happened a lot faster, because for a lot of these vaccines, people were previously actually working on these viral vectors. So the Oxford AstraZeneca one, for example, the technology that the vaccine is based on has been in use for other vaccines as well. They've been testing this same vaccine technology for Zika virus, as well as influenza, the normal influenza shots, as well as in cancer therapy, actually. And so because of that, there was safety data that was there previously. So these mRNA vaccines have been around in clinical trials for over 10 years. So because of that, there was safety data that was available on these viral vectors before. And so the only thing that was different was they would just add the spike protein of coronavirus, which actually in itself does not have any safety issues. And so as a result of that, the safety phase was actually quite fast, because a lot of these vaccines are actually repurposed vaccines from, interestingly enough, the HIV field. And so because of the safety work that has been done previously, we were able to know that the vaccine vectors were actually safe for use and could be used for SARS-CoV-2. So I will move on to the next slide, which is on how actually vaccines work. So what this picture is depicting here is that, you know, we have these different mechanisms that will bring immunity to an individual. You have your protein vaccines, you have your mRNA vaccines, and then you have your live or attenuated vaccines that can give you activity. And so just for us to understand and for us to be all on the same page, I will briefly talk a little bit about what immunity is. So immunity really is the concept which we speak about, in which an individual has almost like a force field that prevents invaders, which are the pathogens, HIV, SARS-CoV-2, polio, from coming into your body. I love this picture. It's one of my most favorite pictures from Black Panther, which shows this shield that actually hides Wakanda from everybody else, you know, which also stops invaders from coming into Wakanda, and it opens up, you know, once in a while to allow people who are allowed into Wakanda only, but everybody else isn't allowed. So this is what vaccines actually do. They form a force shield almost that prevents diseases from coming into your body and causes sicknesses. And so what they do is it's a mechanism that stops these viruses from being able to make you sick, so that when you're exposed to them in the environment, they will not make you sick. And there are various components that actually confer immunity, and there are, you know, this is the only jargon that I think I'll use today. We'll talk with the T cells and B cells and antibodies. These are the cells that actually form that immunity. These are the soldiers of the body that give you that force shield, that come together and, you know, make that force field, so that when coronavirus comes, they actually protect you from that. So what you see is you have your antibodies, which a lot of people talk about. Your antibodies come in and they bind the coronavirus, so the coronavirus cannot infect you if you have antibodies, that is. And you have B cells, which then make those antibodies, which then also protect you from SARS coronavirus. And you have T cells, which are also involved in this. And so what all these cells that reside in your body, what they do is that the minute that coronavirus comes to you, they will then protect you. They will destroy that coronavirus once it enters your body. But the important thing is that these cells are not there naturally. They need to be taught how to do that. So it's almost like they need to go to school. They need to go to university to be told, you know, firstly, how to identify coronavirus, and once they identified, how to destroy it. And so this schooling process is the vaccination process. So vaccination actually teaches your body what cells, what invaders to destroy when they come to you the next time. So these are very important parts. And there are different ways of schooling your body in how to actually destroy these invaders, how to confer that immunity, in other words. And in order for us to understand that, I'll just like to remind us that we have in our bodies RNA and DNA and protein. DNA is what makes up, that's our genetic code. That's what confers, who you are. It determines your hair color, your skin color, it determines certain things such as your height and that sort of thing. And your DNA is who you are. But your DNA actually makes proteins. So your skin color, for example, is due to proteins that are in your body, right? Your pigmentation proteins, your melanin. So they are told to be made by DNA. The message comes from DNA to make those proteins that give you certain characteristics. But in between those is a messenger, which translates that, which is called RNA. And so what these vaccines actually do is there's some vaccines which are DNA based, and some of them are RNA based, and some of them are protein based. So there are different ways to school your body, which is to school these different immune cells on how to identify and destroy those pathogens, in this case, the SARS-CoV-2 pathogen when you encounter it. And they are either DNA, RNA, or protein based. And so I will talk a bit about the genetic vaccines now. Perhaps maybe I should pause a little bit and take one question if there's a burning question. I'm halfway through my slides at this stage.
There's many questions, but I'll give you two. Yumna Hattis asks, how many documented strains of COVID-19 is there, and how does immunity to them work?
Mm-hmm.
The second question is from George Owino, who asks, I would like to know why some of the vaccines have had fatal effects on older populations. Were they not tested among older people? Would you like another one, Doc?
Yes, I can take another one.
Be the charm. Karim Karasise asks, does the vaccine have any kind of special effect in patients with blood disorders like thalassemia or sickle cell disease?
Okay, so those are really great questions again. I mean, wow, this is very interesting. The first question I will speak about later I've got some slides speaking about the strains and vaccines. And then the issue of the fatal effects on the elderly, that has to do with, I think we've all seen that, that reports of the elderly people who unfortunately died after vaccination. And now that one is still being investigated. I mean, that was very seriously taken, these individuals who were given this vaccine. The current thought on that is that, vaccines are, it is a challenge on a body. So vaccines typically are given to healthy individuals. The current thinking, and I'm speaking here in the sense that this has not been finalized, the final reason has not been published yet, but it seems currently that these individuals who were actually given this were already frail and elderly. I mean, so there were people who were very, very ill when they were given this. And the vaccine trials that this was done in, vaccine trials are done in individuals who are not frail. And so the safety is shown in individuals who are healthy. So even though they're elderly, I mean, you have people who are elderly and healthy, and you also have people who are elderly and frail. In this case, it seems that perhaps these individuals were actually already frail. And so, there's a question on whether or not they were eligible for this vaccine anyway. But that story is still being unfolded, and I don't know what the final verdict on that is, and it is very concerning, and people are actively working on that. But there were thousands of over 65 and over 40-year-olds who have been tested with this vaccine who have not had the same effects on them. And so there seems to be something that was happening with these particular individuals who were given this, which is under current investigation right now. And the question on people with blood disorders, my current understanding on this is that there hasn't been any issue that has been shown for people who have blood disorders. The current information that I've come across is that it is safe in individuals like that. If you would like to pop me an email in the chat there, I can verify that for you, but I have not seen any indication of any data showing that people with sickle cell anemia, for example, have issues with this vaccine with any of the vaccine candidates that have currently been developed. So it is being given to people with those conditions currently. Yeah. And so with that, maybe I will speak about the types of these vaccines. So we sort of have an understanding a little bit of what RNA, DNA, and protein is. And so of these different vaccine types, we have the genetic ones. And these are the ones that have been causing most concern. And these are the ones that have been licensed first. So these genetic ones are the ones that are RNA-based or DNA-based. So if you remember in my previous slide, I showed that DNA and RNA are the code that tell your body, that define who you are. And so people have been very scared of this because once you say the word genetic, people get very anxious. They think they're being cloned or they're being modified. People think of GMOs, and these vaccines are actually not that. They do not alter a person's genetic material in any way. In fact, with these RNA vaccines that are being used, the RNA itself that is being injected in the vaccine itself does not even stay active. Your body itself breaks it down after it has coded for the proteins that you need to get your immunogenicity. So these RNA particles that are actually injected do not actually last very long in your body. Your body itself deals with it, but they last long enough for you to get immunity. And so we have 36 that are RNA-based vaccines that have been that are currently in clinical studies, and we have 26 that are DNA-based. The DNA-based ones tend to be more robust. The RNA-based ones tend to be the ones that need this very extreme storage because RNA itself as a molecule is not very robust. It breaks down very easily. And as a result, that's why, they are so difficult to store. But the DNA ones tend to be more stable, but they do not do anything they do not alter the DNA of the person themselves. Which is important. So you will not now start becoming something that you're not because you've taken a DNA or an RNA-based vaccine. What happens is that some of your cells, your immune cells that need to see the message that this virus, next time it comes into your body, you must fight it, get the message enough, but they do not alter your own DNA as they do this. So what a lot of these RNA vaccines do is they actually code for this virus spike protein. So the coronavirus has these spikes outside, and it's called coronavirus because it looks like a crown, and those little projections that it has on the outside are called the spike protein, and those are actually what the RNA vaccine code for. They code for the spike protein so that the next time a coronavirus comes into your body, it will be identified because the vaccine would have taught your cells that if you see the spike protein, go ahead and kill it. And so that's what it does. It's almost the RNA vaccines teach your cells that when they next see the spike protein, they must destroy it. And so is the case with the DNA vaccines. And so the current data is actually quite efficacious that we've seen from these studies. The Pfizer and the BioNTech candidate has shown something like 95% efficacy, and it has been shown to be quite efficacious in individuals that are under 65 and also over 65. The side effects that were shown include fatigue and fever and muscle aches. And mind you, the virus itself actually, when it infects you, does cause genetic changes, because what the virus does is it actually comes into your body, and that actually involves it hijacking your cells and making them make more of itself, which is not what the vaccine does. So the vaccine is a much milder effect on you, which is and then so these vaccines have been licensed, as I was saying, in a lot of countries, in Canada, Chile, Costa Rica, Ecuador, Mexico, Panama, and Singapore. And this is the Pfizer BioNTech vaccine that has been already been licensed, as well as the Moderna one, which has also been licensed, which is another RNA vaccine. And there are other vaccine candidates, such as the CureVac one, as well as the Imperial College London vaccine candidates. And there are actually a lot of vaccine candidates which use this mechanism, which are in clinical trials. And then we also have the viral vector vaccines. And these are the viral vector vaccines, such as, for example, the Gamaleya one, as well as the AstraZeneca Oxford one. What they do is they, in this case, like the Oxford vaccine one, they take the DNA, in this case, not RNA, of the DNA code of that codes for the coronavirus spike protein, and then they put it inside a virus that is that people commonly have. And in this case, they've used an adenovirus. And adenoviruses are viruses that get, like the common cold. Those are adenoviruses. We see them regularly. They don't cause any death in people. And so they take an adenovirus and put this DNA inside it so that when it then is injected in you, your body will know to fight this the coronavirus the next time it sees it. But these viruses, which are the vectors, the adenoviruses, are harmless and are proven to be safe in individuals. And so in this case, we have 35 non-replicating vaccine candidates, as well as 24 replicating vaccine candidates that are currently in clinical trials. And as I mentioned, the AstraZeneca one follows this mode of mechanism, as well as the Gamaleya one, the Russian one. And interestingly enough, there's a partnership between Gamaleya and AstraZeneca to actually develop a co-vaccine between the Gamaleya one and the AstraZeneca one that will be given together, the two of them. So there are quite a couple of candidates that are being investigated in this regard. And then the other types of vaccine that are there are the protein-based vaccines. Now, these protein-based vaccines are the ones that we're used to. So the RNA ones, for example, it's a new technology. And when I say new technology, I'm not saying new this year. It's new in the sense that it's newer than the other ones. As I mentioned earlier on, they've been in studies for over 10 years, a minimum of 10 years. And this is actually in the cancer field, a lot of people have been using these RNA technologies for cancer therapy. And so they're also now being used in the case of infectious diseases and looking at the SARS-CoV-2. But the protein-based vaccines are the ones that we're used to. So, for example, the flu vaccine that we use, even even the Ebola vaccine, and for some of them, we normally use these protein vaccines, and even so there are a lot of protein-based vaccines that we tend to be used to. And in this case, what they do is they take the proteins, they make proteins that are similar to the coronavirus proteins. And when they make these, then they inject this into individuals. And then because they inject this into individuals, your cells then get taught, as I was talking about, this university that your soldiers go to, your cells then get taught that the next time they see that, the real coronavirus will be able to fight it. And so there are currently 89 protein subunit vaccines that are in clinical trials. And then the last ones that I would like to talk to, which were raised by somebody else previously, are the inactivated or attenuated vaccines. In this case, and in fact, we have vaccines like this polio. The polio vaccine actually is an example that we've been using for many, many years. That is an attenuated vaccine. In this case, what scientists do is they take the virus itself, they kill it, they destroy it, they make it non-harmful, and then they inject it back to you. And when you're injected with this vaccine that is destroyed from being harmful to you, it is enough to elicit an immune response, so that when you now see the real virus, you get a nice robust immune response, which then protects you and gives you protection from an infection. And so there are less of these that are in the vaccine pipeline, and that is because they have a risk with them, that if you have issues with making them inactive, then there are problems. But there are ways that people have overcome this, in that they have found ways to actually destroy these viruses effectively, so that when you do inject them into people, they don't make them sick. And in fact, one of the Chinese vaccines that is being used is one of this that has this method, where they take the coronavirus itself, they inactivated and then they re-inject it into people. And so the important thing to note here is that it is not a vaccine that can actually make people sick. It is a vaccine of the coronavirus where it's injected in people that has been, the vaccine itself has been destroyed. So it cannot make you sick from coronavirus. I mean, you won't get COVID from it. And Sinopharm is using this technology, and yeah, there are a couple of vaccines that are using this technology, as we can see here, with 19 inactivated viruses, and there are 4 that are live attenuated vaccines using this technology. And so just in summary, these are the efficacies of the vaccines that we're seeing. And I'd just like to note that normally we don't see such efficacy. I mean, like the measles vaccine that we see, something like 60% efficacious, but these vaccines are like 95%. You know, this is the Moderna one, it's 95%. The Pfizer, the BioNTech one, the Gamaleya one is reported to be 92%, and the Oxford one is 70%, you know, overall. It's higher if taken at a certain dosage. And so they're quite efficacious, and so they seem to be doing a good job. I think maybe I have time for one question, because I've been speaking a lot, and then I'll do my wrap-up slides.
Okay, Doc, a question that has been lingering here. Emmanuel Bamwine's question. Thank you very much, Dr. Nyaradzo, but my question doesn't the medicine have, my question is, doesn't the medicine have a negative effect to expectant mothers who are HIV positive and the unborn baby itself? This is Emmanuel Bamwine.
Okay. Yeah, so that question that you raised about expectant mothers is a very good one. So, you know, as I've mentioned, there are different types of vaccines. And different ones have been tested, the different ones have been tested differently. So the RNA vaccines currently, the current protocol is that they're not used in pregnant women. That is because they haven't been tested for long enough in enough pregnant women to say that they're safe. And this is pregnant women who are not living with HIV or anything like that. They haven't been tested in those individuals. However, there are women who during clinical trials actually fell pregnant whilst they were on the vaccine trials. So when the vaccine trials were designed, they were designed to exclude pregnant women because you don't want confounders, you know, like, you know, if an individual is pregnant, that's a confounding, you know, that could make, you don't know what's happening, is it because they're pregnant or not? So they were actually excluded, but it so happened that there were some women who fell pregnant during these clinical trials. And in those women who fell pregnant during the clinical trials, there has been no toxicity that has been noticed for the woman or for the children as yet. But there were, like what I understand now, I understand there are only about 18, so there really aren't enough to categorically state that it is safe in women that are pregnant. And this is the RNA ones that I'm talking about. So the current view now is that pregnant women will not be vaccinated with these RNA ones, but that is currently under review. And it is under review because the safety data has actually shown that the vaccine itself doesn't cross the placenta, so it doesn't get to the baby, and that pregnant women have not, you know, the ones, like I've been saying, the ones that have fallen pregnant, and also in the animal studies that were done, there were no adverse events in those animal studies. So there weren't any, there aren't any that have been reported currently, but that data is actually very seriously under consideration right now. But the current workup is that generally pregnant women will not be given the vaccine. And that is why it is important that people who are not pregnant and men take the vaccine so that they can protect those people who cannot take the vaccination because it hasn't been tested in them as yet. And so, yeah, there is no way that actually that I'm aware of that it has had any negative effects on pregnant women. So it is actually quite important to vaccinate a large number of people for that reason, which is actually a nice segue to my next slide, which speaks about this concept of herd immunity. And so, as I was saying that, you know, there are some individuals in our populations who will not be able to take the vaccine because perhaps they're too ill. And, there are also individuals who have extreme autoimmune diseases, who have extreme allergies, not allergies like an allergy to nuts or an allergy to, to grass, that kind of thing, but people with extreme allergic reactions, it's been indicated that they should not take this vaccine. And so for the sake of people like that, it's a good idea to vaccinate people who can be, because then they will also confer protection to those individuals. So we've seen that. So with this concept of herd immunity, so maybe I'll just define what herd immunity is. So herd immunity, and a lot of the times when people talk about herd immunity, they talk about R naughts a lot. And so R naughts is the number in which how many people one person can infect with. So with SARS-CoV-2, we know that generally one person can infect 2 to 3 people. So if one person infects 2 to 3 people, we know that it means that we need 60%, 67% of the population to be vaccinated before we can get immunity. So what this diagram sort of shows here is that if one person has SARS-CoV-2 and we only have a few people with immunity, a huge population can actually, the rest of the population will get SARS-CoV-2. But if we have one person in this case who is infected, but all these people have been vaccinated or have immunity, we will only have 2 people who will be infected. And so this herd immunity is when the disease will not spread very far and the whole community is safe. And so we need this herd immunity. And so there are 2 ways of getting herd immunity. You can either let the infection carry out its course and then wait for people to get immunity because they've been infected before. And this is like the Swedish approach, where they've said, well, let everybody get infected, and then once everyone is infected, then we'll be okay. But unfortunately, what we're seeing now is that actually, as this paper shows, which was actually published here in South Africa, is that people who've been previously infected with SARS-CoV-2 different strains, looks like they can be infected with the different strains. So these current strains that have been going around, like this 501YV2 strain, actually looks like it can infect people who have had COVID before. What this was, it seems like that because, the antibodies from people who had previously been infected with SARS-CoV-2 could not stop this new variant. Now, the caveat for this, of course, is that it's not only antibodies that are involved in immune response, there are other cells involved there, but currently it does seem like actually there's a possibility for a reinfection. So the best way to get herd immunity is vaccination, actually. And we've seen now that various vaccine companies are adding boosters for these variants because there's a possibility that these variants that are coming up might not be able to provide immunity. This is sort of as a backup. Now, what are these variants? And I'm going pretty fast now in the interest of time, but what these variants are, it's, viruses normally mutate naturally. So this is a picture of what the coronavirus looks like with those spikes in red that come up there. And these spikes are coded for in the genetic material of the virus itself, which sits in there. And this is sort of what it looks like. So in this spike protein region, in the RNA that encodes for the spike protein, there are some mutations which make it bind more efficaciously to human beings. And so because of that, these variants like the B117 lineage that has been circulating in the UK, which has the same mutation as the one that we also find circulating in Africa, in Brazil, and also in Japan, this mutation makes the virus able to bind more closely. And because it binds more closely, it transmits faster. And because it transmits faster, a lot more people are being infected. So a lot of the second waves are being driven by this virus, which is very transmissible and possibly can reinfect individuals. So it's very important that we get vaccines that are able to deal with this. And this seems to be the case. So the Pfizer and the Oxford vaccine have already been tested in a small number, and they've been shown that they can confer immunity towards these variants. But as we have seen in that other paper, it doesn't seem that people with natural infection can actually do that. So, and this is because vaccines actually give you a more robust immune response. They give you a better immune response than sometimes the normal infection itself. And so, yeah. And just in conclusion, human challenge studies where people have been advocating, to give people the coronavirus itself to see whether or not, and then the vaccine to really see if these vaccines work to speed things up, they are problematic because the ethics of that is there's still so much that people don't know about SARS-CoV-2. We have seen that some people have long COVID, and is it ethical to actually give a person who you don't know how they will respond to the virus itself, to give them this virus and sort of wait and see what will happen? I mean, is that ethical? And what kind of a clinician would actually do that? Because clinicians are there to cause no harm, but this actually potentially is causing harm. So, I mean, there's a lot of discussion on that, but I'm sure you guys know more about this than I do. And then one of the big issues is this vaccine aversion, that we've seen. We've seen a lot of information going around on WhatsApp, these conspiracy theories. It's a conspiracy theories that it's a way to wipe out Black people, and Black people should not take this vaccine, and Bill Gates is putting in a chip, and the beast, and all sorts of interesting things that have been coming up, which are real, which need to be addressed. And one of my jobs as a scientist is to be able to understand from different people what the issue is really, and to provide the evidence so that when you are meeting with your communities, you can actually give them the facts. The only way to dispel some of these is to give people information, and then once we've armed them with information, then it can help them. So we can separate the truth from science fiction and just conspiracy theory. And so we really need partnerships between biomedical scientists like myself, clinicians, and social behavioral scientists, as well as policymakers, to better understand and design vaccines that can work, and in this case to help with the SARS-CoV-2. We know that is one thing to have vaccines, it's another thing to have people taking them, and of course we will need a lot of engagement with a lot of individuals. And so with that, I will stop my talk, and I will allow for questions if we have time, Ponzo. Thank you very much.
Thank you so much, Dr. Nyaradzo, for all your questions, for all your knowledge, and for really sharing. There's lots and lots of questions. I'm trying to go to the top of the Q&As. I've been trying to go between the Q&As and the questions in the chat. So there are questions that we've answered already, but let me start in the Q&A section. Philemon Chuma is asking, kindly, I'll give you 3 questions at a time so you can answer in a batch. Philemon is asking if you could clarify on rumors that vaccines lead to impotence in men. Philemon is from Zambia, and he's from the Resilient Youth for Change organization. Maximina Jokonya says she's interested in the resistance rate among African communities. Do we have any information yet for the resistance around among African communities? And the third question, Dr. Nyaradzo, will be from Adam Mukushi, who's asking if there's any documented cases of efficacy and safety of vaccines with people to people with disabilities.
Okay. Okay, so on the question on impotence in men, so there haven't been any documented reproductive issues in men or in women from any of these vaccine trials. As I mentioned earlier on, these vaccines, the molecules that actually get injected to an individual, they're actually short-lived. They don't live long enough in your body to be able to have an effect on your reproductive system. It's actually one of the criteria, that people look at when they're looking at the phase 1 to make sure that, factors such as sperm production, as well as issues with, reproductive health for women are looked at. So those are taken very seriously. As well as issue, so there aren't any issues of any reproductive nature that have come up at all, and they are taken very seriously during the safety studies. And with regards to data on resistance in African communities, I would love to find that out. I don't know if anyone actually has taken this issue up and studied, you know, what those issues are. I know there's a lot of information circulating around African Americans, for example, with their vaccine hesitancy, you know, because of issues with the violence of, you know, science and on Black bodies, for example, you know, with the syphilis studies and Tuskegee and all of those issues. So that there've been studies there, but I haven't seen or heard of anything being done in Africa. Perhaps the next speaker will actually talk on that. I think she might. But I'm not aware of any of that, but that is definitely needed. And the last question was on you know, vaccine studies with people with disabilities. So, you know, disabilities are very different. There's a wide range. I mean, there are people with mobility disabilities, and those, of course, would not have an effect on individuals because, you know, as long as the immune system and the other biology of the individual is not compromised, their biology is exactly the same as someone without disabilities. But where it comes to some disabilities which are linked with genetic, which are conferred because of genetics, now these are completely different. And those individuals currently, there's a different workup for that. And the individual would need to speak to their practitioner on that. But that's a very specific subset that we're talking about. But generally, people with, you know, disabilities that are not genetic and do not have other physiological aspects, you know, on the person, there has been no issues with that. I hope that answers your question.
Thank you so much, Dr. Nyaradzo. There's more questions. There are more questions that I'll go through now. I'll tell you who they're from and just read them out. Raymond Mazambe asked, what vaccine is recommended for us? What vaccine is being recommended for us African people, especially the mRNA type? There's also a question from George Owino. I would like to know why some of these vaccines have had, oh no, you've answered that, my apologies. I see most vaccines explained by Dr. Nyaradzo are produced by countries outside of Africa. Do African countries have the ability to produce and manufacture their own vaccines? And the last question is from Emmanuel, who is, no, he's Bella Akagba, who's asking, to make sure, no, this is Dadirai who's asking to make sure that the persons receiving the vaccines are not infected already. Are they going to be tested? If they are negative, then they get the vaccine. How does it work? I'll leave it there for now, Doc, until the next round.
Okay, excellent. So Dadira, yes, thank you for your question. So the current protocol is that people who have had COVID before should be vaccinated. And the reason for that has to do with those strains that I was trying to explain earlier on, that we have seen that, well, you know, like I'm saying, a lot of this knowledge is incremental, so we haven't come to the full knowledge of it yet. But right now, it seems that previous infections, especially asymptomatic ones, and of which the majority of people have had asymptomatic COVID, they don't seem to confer protection against certain variants. So if you're living in a country where these deadly variants, well, not deadly, I must choose my words correctly, these variants that are very virulent, that are quite deadly, the easily transmissible one, like the B117 and the 541 in South Africa that we're seeing, for example, you want protection against that. So you definitely want to vaccinate individuals who have been infected before. It cannot harm you. It will just boost your immunity. So that's the current protocol. Definitely people who have been infected before should be vaccinated. And the question on which mRNA vaccine is recommended for individuals in Africa. So all of them. They are all safe in people of African descent. And as I mentioned before, and for me personally, this is one of my little pet passions, that it is very encouraging that actually a significant number of people in Africa were tested. Wits University in Johannesburg has tested this vaccine in sites in Johannesburg, in Cape Town, and in other parts of South Africa. So we have tens of thousands of people in Africa who look like me, who've tested this vaccine and have not had any adverse events. So they are safe for people who look like me. In fact, I myself, because I believe in these vaccines and their safety, have taken one. I have had both doses. I am perfectly fine. My husband also has taken one. No problems in any of those, in any issues. So there is no problem with any of these vaccines for Africans. So either of them is a good choice for a person in Africa. And the last question was on vaccine production. Now, this is another pet question of mine as well. I'm embarrassed because we have so much skills in Africa. There are many individuals in Africa who actually can participate meaningfully in making vaccine candidates. But unfortunately, we have none that are coming out of Africa. And it is very sad because we have no excuse. In fact, a lot of these companies which are producing these vaccines are using skills from Africa. There are lots of people who are at Pfizer BioNTech who actually come from Africa. In fact, so we have no excuse. We should be able to make these vaccines in Africa, but we're not. And a lot of that has to do with cost or policy. So I would love, if there are any of you who are available, who are there, who can advocate for our African governments to actually set aside money for us to do things like that. The knowledge is there, the ability is there, we just need the money, we can totally do this. And then we won't have these issues where we're finding we're last in line for vaccines. So yes, we should be producing vaccines in Africa.
Thank you so much, Dr. Nyaradzo. I see Emmanuel's hand has been up a couple of times since we started, so I'm just going to allow him to verbally ask the question. Emmanuel, you can unmute yourself and ask Dr. Nyaradzo your question. While Emmanuel unmutes himself, Dr. Nyaradzo, I'm just going to go through another set of 3 questions. George Owino asking a question around, sorry, did you talk about the approaches such as BNAbs, broadly neutralizing antibodies? George also asking, was the Moderna one discontinued? And thank you Pumzile Singutu saying, thank you very much for an informative presentation. Currently, there is a call to start clinical trials involving the use of ivermectin to treat COVID-19. Have you managed to get the data or indication whether it can be effective against COVID-19?
Okay, Emmanuel, have you unmuted yourself?
He seems to have not unmuted himself just yet. So I think you can go ahead and answer the 3 questions that I just gave. Oh, there he is. Emmanuel, please feel free to ask Dr. Nyaradzo the question.
Yes, good evening, people.
Good evening.
This is Emmanuel from Uganda, CEO, Hands of Africa. My question is, she has been mentioning, that doctor has been mentioning that vaccine being discovered in most of European countries, but here in Uganda, they are almost in the edge of vaccinating people, the medicine of COVID-19. How quantifiable is that medicine? Has the team from World Health Organization or UN approved this medicine or not? Or it is a team from Uganda? I just want to hear your views about it. Thank you very much.
Okay, I'll start with Emmanuel's question and then I'll answer the other 3. So Emmanuel, you raise a very good question, and it is very important that in all of these things, we make sure we use things that are safe, and we need to be able to verify that they are safe. And these vaccines, believe me, have been tested for safety, and they are approved by the WHO, which is independent of the companies themselves, which actually develop these drugs to make sure that they're safe. And then secondary to that, then the countries that they become licensed in also make sure that they're safe. So by the time you in Uganda are given this vaccine, it has been through at least 4 independent bodies to make sure that it is safe for use in people. So the WHO in all of these vaccines that have been developed has assessed the safety and the efficacy of all these drugs. So we do not take the word of just pharmaceutical companies to say that, yeah, it works and it's safe, but there have been independent bodies that have actually assessed this. And in addition to that, a lot of these vaccines have, well, in particular, the Oxford one has published their clinical data. So it is publicly available for people like me or anyone who wants to actually assess that, is this really, really safe? So yes, they have been assessed that they are safe. And so you can be rest assured that before anyone in Uganda is given this, it has gone through all the safety channels, including the AU itself also assesses safety. So we have bodies that assess this. So yeah, that is definitely an important question, and it is being dealt with. The other questions that were raised were on the broadly neutralizing antibodies. Yeah, so that's a really great question. So there have been studies, especially in the beginning of this pandemic, which were looking at antibodies to help. In the beginning, there was a lot of work on that, where people were even donating their blood to extract antibodies to give to people who were severely affected with COVID. It sort of took off, but not very much. So it has been shown that yes, antibodies from people who've been previously infected can help, but it doesn't seem to be able to help enough and to be able to be practical for everybody to get blood from someone. It has a lot of issues with it. So a lot of work is going towards these vaccines rather than the broadly neutralizing antibodies, but that is still being investigated. And long term, I think we probably will find a lot more therapies that could use that route of mechanism. But currently, with the time that has been there, it really isn't available en masse to a lot of people. It has been investigated, I know, in the US. People in New York investigated that, I know, but it's not publicly widely available to anyone and everyone, that sort of thing. And the Mrna vaccine has not been discontinued. The Merck vaccine, there are 2 candidates from Merck which have been discontinued, but the Mrna one is the one that has actually been licensed in a couple of countries. It is one that requires refrigeration at 8 degrees, not -80, and I think -20, but can be stored at lower temperatures for a bit longer. So no, that one has actually been licensed and is being used for emergency use. It's been licensed for emergency use. And on the issue of ivermectin, I won't speak as an authority on this because it's not my area of expertise. But what I have seen in terms of the paper that was published that speaks about ivermectin, it was not a clinical study. It was, how people go into the literature and they look at old literature and then they do a meta study. So it was not a clinical trial that actually looked at the efficacy of ivermectin. So ivermectin is an antiparasitic drug that is used in animals. In South Africa, for example, it's not licensed for use in humans. People have been asking for it. So currently, I think it has been, the South African health authorities are allowing certain practitioners who really feel motivated to do so, but under strict conditions. But the pharmacokinetic data that I have seen that comes from ivermectin is that the dosage that is needed for it to be efficacious is actually toxic for individuals with severe COVID. So to be honest, it doesn't really seem like it is. It seems almost like a hydroxychloroquine story, but there are studies that are assessing its safety that are currently ongoing. There is no evidence from clinical trials right now to this date that show that ivermectin prevents severe COVID. If anything, ivermectin does help head lice and scabies, indeed, but its use for COVID has not been tested. There was a very small study, I think in 24 individuals, which is nothing. and it was not controlled in the sense that there wasn't a good control group to really say that this drug is what made people better. In the case that if you give 24 people, you say, oh, no, I've given 24 people and half of those got better. I mean, half of individuals with COVID, in fact, more than half get better by themselves without being given a drug. So before you can actually say that a drug is the cause for this recovery, you need to have a control group that don't have the drug and with enough numbers to compare people who have it and people who don't. And those numbers just don't exist for ivermectin currently. So yeah, I think right now there just isn't data that is convincing to say that. And I understand a lot of people are taking it as a prophylaxis, very risky. But yeah, there's no data currently that says SARS-CoV-2 infections are helped by taking ivermectin.
Thank you so much, Dr. Nyaradzo. We also have two people who would like to ask questions. So I'll ask Mrna to unmute their mic and ask the question, then followed by Wilson. Mrna? Wilson, go right ahead. You've unmuted first. It's okay. You can go right ahead.
And Dr. Nyaradzo, it has been a very good afternoon and we've enjoyed your presentation. I still want to get back to the chronic kidney disease because on that one it was silent. Because I'm taking it from the practical experience. I have a congenital polycystic kidney disease. And it so happened that some 4 days ago, I was switched from the losartan that I've been taking to moduratic. I only realized that moduratic, one of the active ingredients is something that has got some adverse repercussions on my kidneys. I had to go back because I'm not supposed to take hydrochlorothiazide. I went back and told the clinician to say, this is the wrong drug for me. So that's why I'm actually being particular, I'm actually particularizing to look at what effect that would have on people like us with the congenital conditions that we were born with and we cannot change it, if we were to get the vaccine. Yeah. So I don't know if there's that has been done to interrogate that before when they were doing the trials on human subjects, or I don't know if they used guinea pigs or mice. What were the results like? Yeah. Before I decide to get the vaccine.
Yeah, that is a very, very good question. And I'm not qualified to say to you that you can actually take this, because this is something that, your clinician who's been taking care of you will be able to assess for you. So what you would need to do, sir, is to talk to your clinician who's been prescribed your other drugs, who has a good knowledge of your history, and understands your condition well, and then to ask them what their thoughts are on this vaccine. I am happy to, if you put your email address in the chat, to look up specifically this specific condition that you're referring to, that I'm happy to share with you, which you can share with your clinician. But your clinician is the best person to advise you on the safety of this, because this is a very specific and almost like niche question, which there isn't a blanket answer for, because like I said, vaccines generally are tested in individuals who are healthy and don't have some of these congenital issues. And so those would always need to be addressed on a case-by-case scenario. So yeah, so please do pop your email address and I'll send you whatever research I have on this specific issue with regards to vaccine, which you can share with your clinician, which will be able to help you.
Thank you so much, Dr. Nyaradzo. Again, I will ask for Mrna Msongo to unmute their mic and ask their question themselves. Mrna, if you can hear me, Mrna, if you can hear me, please just unmute yourself and you'd be able to ask Dr. Nyaradzo any questions you may have. as we wait for Mrna to unmute themselves, I'll read I'll say 4. We're left with 8 questions, so I'll read 4 now and take 4 later, Dr. Nyaradzo. Samuel Painstill says, AstraZeneca vaccine from the late stage trials has been found to be 70% in protecting against symptomatic disease. It's, however, stable at higher temperatures compared to the others. My question is, with the reduction in efficacy, will it have a negative effect if used during the pandemic, especially in African countries, where people might think because they've been vaccinated, they might not follow protocols, which is already poorly adhered to? That's the first question. The second question is Yumna asking, can people with Hashimoto's disease be vaccinated? Another question from Damali Naigaga, should we trust vaccines that are developed especially from African countries? I'll leave you with those 3 questions and then we'll do the last few. And I see Mrna has just confirmed they've put their question in the Q&A, so I'll get to it. Thank you so much, Dr. Nyaradzo.
Okay. So the last question was on, can we trust vaccines that have been developed in Africa? When we do have them, because currently we don't have any, we can totally trust them because they will go through exactly the same safety and efficacy rigmarole that any vaccine developed from any country will have to go through. And there are a lot of able and dedicated scientists in Africa who are very able to do that. And we have very able African talent that we've exported as well. So anecdotally, the BioNTech guy, one of them actually, who was in the press recently, not the BioNTech guy, one of the Moderna senior individuals is a person who went to school here in South Africa. And we know, with just how many people we export from Africa to the West, that I can guarantee you in all of those countries, in all of those facilities, we have a lot of African talent that is there. So believe you me, there are already Africans who are involved in this, who have been involved in this, who have done amazing, great work. And if we had a platform to do so in Africa, I would totally be 100% sure that we would be able to. In fact, there are some platforms that were involved in that, like the BioVac Institute here in South Africa. And one of the deals actually that hopefully Johnson & Johnson is getting into with Aspen, which is a South African company actually, which will be involved in producing some of the vaccine if it gets licensed, if it goes through if it gets approved, and if the deals go through, some of that might actually be produced in South Africa, which will then make it easier for the rest of Africa to access, if that is actually the case. So that will be actually a positive thing for us in Africa, because it's easy for something to get to Uganda if it's coming from Johannesburg than it is to come from wherever else it will be coming from. So definitely we need African vaccines. We need African-made vaccines. And the question on Hashimoto, I'm afraid that's also another question I'll have to revert back to you, because I do not know that, and I should actually, because there are a lot of people who live with Hashimoto. But I will actually get back to you on that, because I cannot say right now. If I do, I would be lying. And the issue on adherence of vaccination. So because, and unfortunately, as we've been saying, right now we don't know still if people who have been vaccinated, whether or not they can be asymptomatically infected, and if they can then transmit. So the current protocol is that people who have been vaccinated still should wear their mask and adhere to those non-pharmaceutical adherence protocols, which I know there'll be another talk on later, which can allude more to that.
Thank you so much, Dr. Nyaradzo. I know we should be wrapping up, but clearly you are, very popular with many, many questions coming my way again. I'm going to combine 4 questions again, and then after you answer that, we'll take 2 participants who would like to ask you questions directly, and then we'll take the last batch of questions. Balaba Jacob wants to know, does everyone who has been tested on does everyone have to be tested in every type of vaccine? If so, how long does it take for the second vaccine to get the second shot of the vaccine after another? He also asks if children under the age of 6 years are able to participate in trials. I think you've touched on that, but you can just quickly answer that again. Edith Kamau from Kenya is asking, is there any expected COVID vaccine side effect, particularly for people living with HIV? Where Rema's question is, can you kindly revisit the page where you defined immunity? I'm not comfortable with that explanation. So, and Rema, you can write in the chat exactly what you're not comfortable with so we can share that with Dr. Nyaradzo.
Okay, sure, there were a couple of questions there. So, yeah, on the, well, okay, the immunity one, I think we'll find out more about that. Yeah, Ponzo, maybe do you want to, because I was reading some of these, so I was reading the chat group on some of these questions there.
Yes.
So was the one on the PKD that you were asking on? And so, I mean, I think Wilson Zulu asked that. I think
yes.
what I would like to do again is, like, with a lot of these, I cannot comment on them because those are questions that a clinician who knows the condition and understands you can best answer. So I unfortunately, I'm not qualified and I'm not the best person to do that. And if anyone wants to tell you otherwise, they'll also don't trust them. Go to your clinician who'll be able to tell you. So with the question with regards to the different vaccines, if everyone will need to be tested with them, unfortunately, how this will actually work out, and I think there will be somebody who will be talking on policy of the vaccines later on. How it will work is different countries have made arrangements with different vaccine manufacturers. So, for example, South Africa has made a deal with the Oxford AstraZeneca guys through the Serum Institute in India to get vaccines. So South Africa will be vaccinating its population with that. So even if I wanted, for example, the Novavax, I will not be able to get it because my country of residence does not source it. And in our case, the government is the one that is procuring vaccines. So even if I might want to take a different vaccine, I'm not able to because it's not available for me in my country. It's not like I have a whole, cocktail that I can choose from. In fact, at the end of the day, what it will be for most of us is that we will be only able to take what our countries have decided on. So it is important for you policy makers and advocates to advocate for our governments to source the different vaccines that we in our communities actually want. So I think we should totally do that if we can. If you find that in your communities people would rather not take the genetic ones, then ask them to source the ones that are not, and because those options are available. So that's actually how it will work. So each person doesn't get tested on the different vaccines. You literally will end up taking what is available in your country. And then I think that was the other is the one question that I haven't answered, Ponzo?
Yes, there's a question, I'll get to it just now. It was around, are there any expected side effects, particularly linked to people who take the vaccine who are HIV positive?
Yes, so people who are living with HIV, who are on ARVs, have participated in these clinical trials, as I've said. There have been no adverse events in people living with HIV, which has actually been very, very good for us in the South because we have huge communities who are living with HIV. So people who are living with HIV should definitely take the vaccine. It has not been shown to be poisonous or toxic for individuals like that. So it is a safe vaccine. And the other question was on children. And like I said, also with pregnant women, this vaccine has not been tested on children. So currently, it is not being offered to children, but the next phase of clinical studies will test it in children. But before they go to children, they'll test it in adolescents. But that is the next phase. They haven't gone to that stage yet, and we're all waiting to find that out. But currently, they haven't tested in children or pregnant women. So it is not being given to pregnant women or children for that reason. Yeah.
Thank you, Dr. Nyaradzo. And I think one last question, which I think it was also a reiteration of something that you said in your presentation from Dadirai Manyarara, who asked about side effects and whether when we weigh the side effects against the benefits, does safety still count?
Absolutely. And I think, with everything, that's what it is. I mean, it's medicine, right? There's never any medicine that you just want to take when you're not sick. You take medicine because it has a beneficial effect. So if its benefits are less than the risk, then do not take it. Absolutely. It has to have an overall good. Otherwise, there's no point. So there are adverse events let me qualify that. With anything, with vaccines, you get a prick in your arm. That is not comfortable. It is painful, right? But thank you. I think the discomfort that one undergoes by being pricked in the arm, and with some vaccines, there's some fatigue that you can go through, or some swelling on the site of the injection. Even with some of us with BCG, you get the scar that you remain with for the rest of your life, right? Or those other things. So those are some of the effects of vaccines, but that far is outweighed by getting COVID by any chance, where COVID itself can do so much more harm to you. So the side effects of vaccines are very mild. It's those that I've mentioned. Some people get headaches, some people will get the soreness, and I mentioned I've taken a vaccine. I had some redness on the site, but other than that, that was it. It really was just like a prick. And sometimes, people are uncomfortable with being pricked, but really that is the worst that can happen to you. But the effect of COVID is far much worse.
Definitely. And I just want to tell everyone listening and watching is that, due to the fact that we have another talk, the second lecture for today will start in about 18 minutes. Morifi, Santi, and Onufaith have had their hands up for quite a while. So I'll allow them, and they'd like to ask questions themselves. Those are the last two questions I will take for Dr. Nyaradzo, because we do have to prepare and just make sure we have the next lecture, guest speaker up. And so those are the last two questions we will take. Morifi, I'm just unmuting you. You can please ask the question.
Thank you, Dr. Nyaradzo, for such an informative presentation. So I have a question regarding people who were once infected and who did antibody tests that they found they had COVID-19 antibodies. Do they qualify for vaccination? And I wanted to ask the other question in terms of policy development in the countries. How far is it? Because I know such programs of vaccination could not happen unless there's a policy that stipulates and defines the criteria for vaccination. Can you help me with just those two questions? Thank you.
Definitely. So prior infections, it is important for people who've had prior infections to be vaccinated, even if they've been seen with antibodies. The reason for this is that with an infection, like we were saying, some of the data that has come out of Penny Moore's lab at the NICD here has shown that, for example, like I was saying, that people who had previous infections with a different strain of SARS-CoV-2, when they had this 501 mutant, their antibodies were unable to actually neutralize it. So for that reason, it is important that people, who have been previously infected get vaccinated, because you don't know if their immunity they have can protect them from any type of strain that will come in the future. Strain, I mean variant, I mean. So it is important that people have immunity towards all types of variants that can come. And so that is why vaccination is important, because an infection cannot confer that necessarily. So yeah, people who have had prior infections need to be vaccinated, and it does qualify them. And that is the workup currently, that it does not exclude you from being vaccinated. And then on the issue with policy development in the different countries for vaccination. So, you know, for those of us in Africa, in fact, everywhere in the world, because of the problem of, you know, COVID-19, most policy has said that people who are frontline healthcare workers should be vaccinated first. And that is because they're the ones who are most exposed. So people who meet a lot of people, so even including policemen who have to, you know, patrol the streets, they encounter a lot of individuals, so they would need to be protected, because if they are, you know, infected, then they're not doing their jobs, and everyone in the country is affected. So different countries have different prioritization, but generally most people are prioritizing frontline healthcare workers, and then people who are working with large numbers, and then after that, people who are over 60 years old, and then after that, the rest of the individuals. So different countries will have different policies, but generally that is the current way things have been going on. But there really is a need for engagement with the public on who should get vaccinated first, for sure.
Thank you so much. And now the last question from Uno Faith. Faith, please do ask Dr. Nyaradzo your question.
Okay. Good day. Thank you, Ma, for your fine presentation. Although it's not long that I joined the Zoom meeting, but have learned so many things. And one of my questions I want to ask is that in my country here in Nigeria, so many people don't want to take the vaccine. They say they will not take it because of the past things that have happened. So my question now is, since COVID-19 have this different strand, I'm asking how possible will the vaccine help, because they have the friendship and some people who are not yet infected with it. But what about those who are infected with it? Are they going to give them the vaccine? And if people are having different strand, how possible? How will this vaccine cover others? So I'm confused about these things. Thank you.
Sure. Yeah. So, you know, maybe let me help, you know, a lot of us to understand this issue of the strains. So variants, let us call them variants, because right now that's what they are, they're variants. So there will be different variants, and they rise spontaneously. When you get a vaccine, when you're vaccinated, in most cases, you're protected against a large number of variants. The best current, the most surety we have of being protected against the different strains is from vaccination. A previous infection does not necessarily protect you from all the strains or the variants. It will sometimes help you to not be infected by the same variant. But sometimes, possibly, you could be, if you did not have a robust immune response to the first infection, you technically could be, you know, if you encounter the same strain, get it. And so, because that's the other question that we don't know how long immunity, natural immunity to SARS-CoV-2 previous infection lasts. You know, like in the case of the flu, you know, you get the flu every year. Every time, every season, the flu comes, you get it over and over again, because immunity towards influenza does not last a lifetime. But we know, for example, with mumps, once you get it, you never get it in your lifetime, because that immunity is long-term. So with SARS-CoV-2, we don't know enough yet if natural immunity will confer lifelong immunity. The current indication we have is that the best bet that individuals have for long-term immunity and the most coverage against various strains is, various variants is through vaccination. So that is why we're saying that even people who have been infected previously and have recovered, they should be vaccinated for that reason. And so people who have a current infection of SARS-CoV-2, they need to wait after they've recovered from that to be vaccinated because they're unwell. So the vaccine is not given to people who are currently ill, generally. They wait for the people to recover and then they give them generally, but there are always caveats to that. So, Onu, I hope that helps you in Nigeria. And I understand, yes, there's a lot of hesitancy amongst people who don't want to take the vaccine. But I think it is important to remind people that, diseases like smallpox, like polio, don't exist now because of vaccinations. So we need to inform our communities that actually vaccinations have been around for a very long time. This SARS-CoV-2 vaccine is a new vaccine, but vaccines have been there for many, many, many years. And because of that, a lot of people have children which live up to old age. For example, we know that our grandmothers and our great-grandmothers whose children were not vaccinated, a lot of their children died in infancy. But these days, most women can raise their children to adulthood because of the effect of vaccine. They're protected against smallpox and all those other diseases because of vaccinations, against polio. So vaccines definitely save lives. Vaccines definitely have a good public health effect. And this SARS-CoV-2 vaccine will do exactly the same for us. It will provide protection for us. And we just need to inform our communities and give them the information and engage with them and dispel those myths that are there, because the science is solid. They do save lives. I hope that answers you.
And just like that, our first lecture is done. Dr. Nyaradzo, thank you so much for such an informative lecture. I learned so much. As a health reporter, you think that sometimes we think we know everything about COVID because we're reporting on it, but I got to learn so much about the science behind vaccines, and so appreciative of your knowledge and expertise. Thank you to all our attendees as well for asking all the questions. As I've said in the chat, as the presentation was going on, we will share all the speakers' slides with you at the end of the series. And I'd just like to thank particularly Dr. Nyaradzo again for such a wonderful session. So we are about 8 minutes away from our next session with Dr. Jennifer Maroa, and please feel free to stay online. You can just mute and take a body break, and we will be back starting promptly at 6 p.m. And if there's any technical glitches, because obviously as we're aware, we are doing some things in the background. If there's any technical glitches, like perhaps you come back from your little stretch break and you find that you have been disconnected, please be reassured that you can use the same link that you used to join this meeting to join it again. It might have been just a technical fault from our end. Anyway, see you in 7 minutes.
Thank you very much, everyone. I really enjoyed myself. I enjoyed taking your questions, and I also have learned a lot. Thank you very much.